guys, welcome to week 14. Here we go, development 1. You know how I feel about this, so we're going to do this quick, we're going to do this fast, and we're going to get through the information as quick as possible. So here we go. So this week in development, we are dealing with human development. Now, this week, next week, and week 3 of development is talking about all the different stages of human development. And during development 1, we are covering from conception to birth. Next week is birth to puberty, and then from post-puberty to death is week 3 of development. So we're going from there. Now, what is human development? It's a scientific study of the changes that occur in people as they age from conception until death. As you know, just in your life alone, how you've changed in the last year, in the last 2 years, in the last 10 years, all those different types of things. It looks at it in a much bigger scale. Now, when we look at human growth, we have to look at it in a longitudinal design. Is one way we can study it, and it's a research design in which one participant or group of participants is studied over a long period of time. So one person, or the Harvard study is probably the most common example that I've been using, is 200 men from Harvard University starting in 1915 or 1918. At the age of 18 were studied until they died their whole life. Every single year, Harvard University would check in with them and ask, and they got to see different changes, transitions. A lot of them went to World War II, all that types of stuff. So very cool stuff that we learned, and that's a longitudinal study. It's a perfect example. Now, another type of developmental design is a cross-sectional design, which is when you're looking at research design in which several different age groups participate and studied at one particular point in time. So when you're looking at a 5-year-old, a 10-year-old, a 15, and a 20-year-old, and they are being tested on Tuesday, and they are doing a bunch of questions, and then they are done, and you will never see them again. That is a cross-sectional study. You're looking at different ages all at one time. Then finally, you have the cross-sequential. The cross-sequential is a research design in which participants are first studied by means of cross-sectional design, but are also followed and assessed by periods of no more than 6 years. So what that means is people of different ages are being studied at one time. Then they are being studied for like 4 or 5 years down the road, and then that's it. So it's not as long as a longitudinal, and it's not as short as a cross-sectional, so it's a nice difference. Now, in your vocabulary, you have cross-sectional and cohort. Please keep in mind cohort and cross are the same thing, so just be aware. Now, here are some examples of research designs of all three. So other examples if you'd like to take a look. Now, some of you have been like, oh my God, nature, nurture again? Yes, nature, nurture again. We will deal with this pretty much on and off for the rest of the year as well. It's a continuing trend. Now, when we deal with nature, it is the influence of our inherited characteristics of our own personality, physical growth, intellectual growth, social interactions. These are all the things that are passed down from your parents. Your genes, your chromosomes, all of those different types of things are what we're inheriting, and that's the nature. Nurture is the influence of the environment on personality, physical growth, intellectual growth, and social interactions. So these are all the things that affect you, that are affecting you from your environment. Like, for instance, your first environment is actually the womb. Now, if your mom drinks alcohol and it passes to you as you're in her womb, that is your first environment. So you will have brain damage because of that alcohol. Now, when we study nature and nurture, we're looking at behavioral genetics, which focuses on nature and nurture, seeing what comes from where, how much influence, all those different types of ideas. Now, when we start talking about nature, we have to talk about genetics. Now, genetics is the science of inherited traits, which describe all the things that make you who you are. Now, when we talk about genetics, we have to talk about DNA. DNA is a special molecule that contains the genetic materials of an organism. Now, this is essentially the recipe of how you are you. Now, a gene is a section of DNA, so it's a smaller part having the same arrangement of the elements. And the genes are what carry your information of what makes you blonde, what makes have blue hair, all those things. DNA, it takes, are you blue hair, brown hair, how tall are you, how your weight, all those different types of things are going to kind of fall into it. Now, underneath genes, we have dominant and recessive. Now, dominance refers to a gene activity that controls the expression of a trait, while recessive refers to the gene that only influences the expression. So, all of those definitely play a part into who you are. Now, here's an example of a gene, the rungs of the ladder that link the strands. Okay, we have all different types of things. Now, when we talk about genes, we have to talk about chromosomes. Now, chromosomes are a tightly wound strand of DNA or genetic material or DNA. Now, the chromosomes are, you get 23 from your mom and you get 23 from your dad for a total of 46. Now, chromosome disorders include Down syndrome, Klinefelter's, Turner's, and genetic disorders, PKU, cystic fibrosis, sickle cell anemia, and Tay-Sachs disease. Now, all of these chromosome disorders are inherited from your parents. They are in nature. They are passed down, and they are given to you from your parents. Now, when we talk about genetics, we're dealing with conception. Now, when we deal with conception, we deal with an ovum, which is a female sex egg that is unfertilized. Fertilization is the union of sperm. So fertilization is when a sperm and an ovum meet, and then they create a zygote. And a zygote is a cell resulting from the uniting of the ovum and the sperm, and that will eventually divide into many cells, which will eventually lead to the baby. So the first stage, it goes ovum, then zygote. Now, once we have a zygote, it could split into identical twins. Now, identical twins are known as monozygotic. Now, monozygotic twins form when one zygote splits into separate masses of cell, each develop into a separate embryo, which will eventually develop into a single cell. So, a monozygotic twin is a zygote that splits into two. Now, dizygotic, typically called fraternal twins, are when two ovums are released by the mother. Both eggs get fertilized, both ovums get fertilized, and they both are turn into zygotes, and they both grow into babies. They have the same genetic matching as I do with my older brother, or you do with your younger sister. You look alike because you're related, but you're not identical. It's just, there's the uniqueness is that you were both born at the same time, but everything else is as similar to everyone else. So identical twins is one zygote splits into two, and then you have two identical babies. Fraternal twins is two ovums are released, two zygotes are created with fertilization, and then you have two separate babies. Now, when we deal with pregnancy, the first step is germinal stage. Now, once we have conception, so the ovum turns into a zygote due to fertilization, we enter the germinal period. Now, the germinal period is about 2 weeks long, and it is during which the zygote moves down to the uterus. Typically, conception occurs in the fallopian tubes and begins to implant on the lining of the embryo. And this is going to start from here to 8 weeks, and this will be the fertilization. Now, where that embryo attaches is where the placenta is going to grow. Now, once the zygote attaches to the uterine lining, it's called the embryonic period. Now, the embryonic period means a zygote has changed from a zygote to an embryo. Now, the period for an embryonic stage is 2 to 8 weeks after fertilization, and during this, the major organs and structures of the organism are starting to develop. You're going to see a big amount of growth. During this time period, you're going to have the critical periods, which are times during certain environmental influences have an impact on the development. Now, there is not a good time to drink during pregnancy. Believe me, I'm not saying that. But drinking during the embryonic period is very, very, very severe. Teratogens are any factor that can cause a birth defect. What we're going to see is drugs, alcohol, any of those types of things are teratogens and that are introduced to the baby that endanger the baby. Now, critical periods, and teratogons. Both of these happen in the embryonic stage. So here are some examples of some teratogons: marijuana, alcohol, nicotine, all of those different types of things. Okay. Now, periods of pregnancy, your final period is going to be the fetal period. This is the longest period. It's also going to have the most growth. It is from about 8 weeks after conception until the birth of the child. This is when the ovum, which turn into a zygote, which turn into a embryo, which turn into a fetus, is now called a fetus. Now, a fetus is a name for the developing organism from 8 weeks after fertilization of the baby. It will be a fetus until it is born, and at that point, it is then a baby. Now, when we deal with birth and all that stuff, the top left photo is a picture of the germinal stage. So as you can see, it's very small, very teeny tiny. Your second stage is about mid-process through your germinal embryonic stage, and then your bottom is going to be the fetal stage about midway through. And that is what a baby looks like in gestation. So anyway, it's definitely an interesting week. You guys had some great questions. I'm looking forward to hearing some more. Anyway, I hope you have a great week, and thanks for checking in. See ya.